Welcome to our webinar on James River Watch. My name is Ben Watson. I'm the staff scientist at the James River Association, and James River Watch is our bacteria testing and water quality monitoring program. Lots to cover today. Certainly a lot about the program itself. I'll also talk a little bit about JRA as an organization. First, I want to thank you guys. I want to thank you for calling into the webinar and also for your help with the program. It's been a success. It's been proven year after year to provide reliable data, and we couldn't do that without the help of staffers of ours across the watershed, and certainly without the help of our volunteers. And I think I'll jump right in. First, we'll be talking a little bit about JRA and the program conceptually, and then we'll dive into field sampling and lab analysis techniques. As I said, James River Watch is a water quality monitoring program, and we look at bacteria levels and more generally river conditions across the whole watershed. There's some website information for you here if you're interested. I'm assuming you're accessing this through jrava.org. There's also jamesriverwatch.org, which is the website more specifically associated with the program. If you have any questions about the webinar, about anything we cover, or any questions over the course of the program this summer, absolutely feel free to shoot me an email and get in touch. You can also give me a call at 804-839-3046. And I've also included some contact information to some coworkers of mine at the regional offices in the bottom right corner. And to give you a little bit of an idea of the structure today, first we'll be going over some background, some requirements and expectations of you all, our volunteers, as well as expectations that you all can hold for us, field and lab techniques, and then how to submit data. We are an environmental nonprofit organization whose mission is to be a guardian of the James River. We protect the James and its tributaries, and we connect people to these magnificent natural resources. And we do this through several core programs. These include restoration work, tree plantings in riparian areas across the watershed, living shorelines in the lower James, education work at a number of our facilities, getting kids out on the water, community conservation, going out into communities and making meaningful impacts that connect people to the river, and last but not least, our advocacy and riverkeeper program, where we talk to representatives, politicians, where we connect you all with representatives and politicians to tell them about the importance of environmental preservation and conservation in Virginia. The reason for the existence of the program is because we receive pretty frequently a very common question, and that is whether or not it's safe to swim in the James River. It's typically safe to swim in the James, but it does depend, and it depends on a number of different factors. It depends on river height, certainly. It doesn't make much sense to swim in the river at flood stage. It depends on temperatures. It doesn't make much sense to swim in the river when it's freezing outside, and it also depends on bacteria levels. Now, historically, river height and flow and water temperature and air temperature, those types of data are very easy to stream. They're very available, and they're easy to get our hands on and compute. But bacteria for a while has been a missing link of sorts. And so that data gap is what this program is designed to address. And also, while we're collecting bacteria data, we figure it's pretty simple and easy to also collect observational data, qualitative things that you see out there when you're taking your sample, conductivity, and air and water temperature as well. So James River Watch is more than just testing for the sake of testing. There's also a forward-facing web app that allows not only participants in the program, but the general public to explore the data that's made available. We hope that it empowers river users to plan their weekends, whether or not they hope to go swimming, boating, paddling, however they want to enjoy the James. We hope to provide them with the data to do it. And we hope to do it in a way that is pretty simple and easy to understand. We update our data weekly over the course of the monitoring season. The monitoring season stretches from Memorial Day to Labor Day. We'll talk a little bit more about scheduling in a bit. But again, the reason the site exists is to address this need that the public has for this type of data. Now, we're not just testing any kind of bacteria. Bacteria come in all kinds of shapes and forms. What we're looking at specifically are E. coli. These are fecal bacteria, which means they are found in the fecal matter of animals, of people, of pets, of wildlife. There are a variety of sources. And as I said earlier, there are a variety of bacteria, but the reason we look at E. coli is because it's pretty easy to test. So really what we're doing is we're looking for E. coli because it's easiest to spot, And if we see it, it indicates that other nastier and harder to find types of bacteria may be present as well. I alluded to sources earlier. They come from people, pets, they come from wildlife, they come from leaking septic systems and antiquated septic systems. So bacteria levels, of course, vary through place, but they also vary quite a bit through time. If it's been dry for several weeks, bacteria levels are more likely to be low. But remember, these sources, pets, people, their waste products are transported after heavy rain events. So just because it rains doesn't mean that the river is necessarily dirtier, but it does make it more likely to be. Now, James River Watch is a program that tests 27 sites across the watershed, but here we're looking specifically at Richmond because Richmond and Lynchburg have a common problem. They're old cities, their infrastructure is old, and their sewage systems are old. They treat their stormwater and their wastewater at the same facilities. It initially makes sense because they have to be treated to the same standard. But the problem with that is that when it rains heavily, these systems are overwhelmed and they have to release an untreated mix of stormwater and wastewater into the river. This does not happen every day. When it does happen, it happens at these red points displayed on the map. And these red points, the first ones to overflow are generally the ones that are a little bit further downstream in the city of Richmond. Conditions will vary day to day, place to place, but on average, the James River tends to have higher bacteria readings closer to the downtown area and immediately below downtown than in some of the parts of the river near Reedy Creek, near Pony Pasture, near Huguenot Flatwater, and so on. And briefly, without diving into too much detail, bacteria impairments are the most common type of water quality impairment in Virginia. On the main map we're looking at here, they're shown in pink. And I know it might be a little bit hard to read these graphs on the bottom of the slide, but if you look into the lower left, what you're looking at are mileages of impairment based on whatever type of impairment it is. And again, in pink are bacteria. They're 3 times higher than the stream mileage, the impaired stream mileage associated with benthics, with dissolved oxygen, and with some of these other less common types of water quality impairments that we see. So James River Watch tests 27 sites located across the watershed. There's a cluster of them in Richmond, but overall they extend all the way from Buchanan in the west down to the Hampton Roads area in the east. Here we're also looking at the average pass rates of these sites, and we can kind of get a sense of some bigger picture trends. Generally speaking, pass rates are a little bit lower on average near urban areas. So the city of Richmond, the cities of Hopewell, Petersburg, the Tri-Cities area, it looks like there's a little green one down by Williamsburg as well. And also I want to point out that on average, if you're in the city of Richmond but upstream of downtown, pass rates tend to be a little bit better than if you're closer to the downtown area. This map is based on 6 years of compiled data available in this table here. And actually both these figures are available in a bacteria report that we've posted on our website. And again, these sites are listed, I believe, west to east, starting with Buchanan, with their historical pass rates and the number of years that we've been sampling at each one. So I am the main organizer of this program, but there's no way I could do it without the help of other staff. And over the course of the summer, information flow is a very important thing to keep in mind. And I'm happy to answer any questions where there's doubt about who to contact. But for day-to-day sampling things, what we've done is with our satellite offices, we have outsourced some of these tasks to the folks who we have present at these offices. I'm out of Richmond and can't be everywhere at once. So for day-to-day tasks happening in the upper, a first contact to consider would be Rob Campbell. His email is right here, rcampbell@jrava.org, and his number is 434-944-7975. JRA recently opened a Scottsville office where we'll primarily be doing restoration work, but we have a restoration coordinator, Anne Marie Roberts, who's there on site. And so for day-to-day issues, you can reach her at amroberts@jrava.org. I'll be covering most of the middle, extending from the Rivanna, where we have a partner, the Rivanna Conservation Alliance, doing good things and testing data that they're sending to us, all the way down to the Hopewell and Upper Chickahominy area. For questions, whether they're small or large, concerning these sites, you can reach out directly to me. And the lower is Emily Cope. She's out in our Williamsburg office, and her email is ecope@jrava.org. Her number is 757-856-1252. Briefly, I'll cover some requirements and expectations. Volunteers are expected to attend one training a year. It doesn't necessarily have to be in person. For new volunteers, it does, but this webinar suffices for people who are already familiar with this program and who have helped us in the past. From a time standpoint, we tell volunteers to expect to take about 3 to 4 hours a week. Most of this is on Thursday, with about up to an hour of time on Friday, compiling the data and uploading it to the website. I'll add that this amount of time goes down as volunteers get more familiar with the procedure. We ask volunteers to test between 10 a.m. and 2 p.m. on Thursdays, and then to come in 24 hours later on Friday to determine their results. If you take a sample at 9 a.m. or 3 p.m., it's not the end of the world. If it incubates for 23 hours, 25 hours, that's fine too. But we do ask that for the sake of consistency, these windows and incubation periods are what we all aim for across the watershed. Again, sampling takes place between Memorial Day and Labor Day, and we ask that volunteers participate in at least 2 or 3 weeks of sampling over the course of the summer. Now, there's a lot of information to distill here. All of that information is included in our River Monitor manual or our standard operating procedures. Those both refer to the same document, and there's a copy available in each kit at each site we have across the watershed. I want to stress again the importance of contact and communication here in determining schedules and who's testing where and when. Absolutely feel free to keep in touch with me and with the other members of your sampling team at your sampling location. But of course, expectations are a two-way street. You all can also hold us to some things. We commit to provide you all with the training and with the sampling gear you need. We have 80 to 90 volunteers each year across the watershed, and we couldn't do it without you, and we're trying to make your jobs as easy as we can. We'll work with you all to determine a schedule for each site that works for each one of you. We'll assign you guys to these sampling teams at each site. We'll share contact information to make sure that if you have an unplanned absence or vacation, which happened over the course of the summer, that we can fill that gap in scheduling with someone else who's available to test. And we also will be very happy to provide you all with the one-on-one assistance that you need if you have questions about methods, the procedure, about uploading data, about anything related to the program at all. And last but not least, the database. This is the forward-facing part of the project where the public can view your hard work. We'll be timely with our review and approval of the data that you submit so that we can get it online for the world to see. And we'll also do what we can on our social media platforms to promote this data, to raise its visibility, and to make sure that it's being put to good use. Okay, field and lab techniques. So I'll be switching out of this PowerPoint shortly, but first I want to briefly cover the types of data that all of this gear we're about to run through is designed to collect. We'll be looking at air and water temperature. We get that in the field. Conductivity, which is also a field measurement. Bacteria, which we'll do in the lab. Turbidity is in the lab, although we need the water quality samples that come from the field, and some of that miscellaneous info as well. A quick note here on bacteria testing methods. For almost all of our sites, we look at ColiScan. ColiScan is the method that uses pipettes and petri dishes and agar, the growth medium, and you count these different colored colonies on plates. Most of you all are probably familiar with that method. We also use ColAlert, which is a more precise method in Williamsburg, and instead of relying on color differentiation, it relies on whether or not samples glow under UV light. So it's a pretty cool procedure, and for more information, go ahead and visit that link at the bottom of the screen. This is the sampling sheet that you all will use to record data. We've broken it down in two ways. One is by what day the procedure occurs on. You can see Thursday and Friday on the left side. The other is whether or not it is an item that you record in the field or in the lab. You can see those upper lighter colored rows. Those are all things that we collect in the field. Let me highlight these for you right here. And then those gray rows below, starting from turbidity on down, those are all data that we collect in the lab. And on this slide, we have an exhaustive list of all the material you'll ever need to run through this whole procedure.
separated into equipment that you'll need in the field on the left side and lab equipment on the right. It's a lot to look at. Pause if you'd like to read through, but I think the best method of going over all this gear is to see how and when it's used.
I'm about to walk through a full round of sampling, starting with picking up the gear and then going to the field, collecting the samples and data that you need, heading back to the lab and processing this data on Thursday and on Friday before uploading it online. First, I want to start with a couple tips. Before you leave for the field, there are a couple of things that you can do to speed things up. 1, take the appropriate number of Collie-Scan agar gels out of the freezer. These take about half an hour to thaw and can't be plated until they're liquid. Most of you will only need one of these each week, unless it's a week where we ask you all to do duplicates for data quality purposes, or if you're in Richmond, where you might be doing 3 or 4 sites at a time. 2, turn on your incubator. When you set your incubator, make sure to set it at 37.5 degrees Celsius. Last but not least, make sure you have what you need. That includes your tackle box full of conductivity meters, air thermometer, gloves, hand sanitizer, both kinds of sampling bottles, writing utensils, lunchbox, freezer pack, phone, and sampling pole. Once you collected everything you need, you're ready to go. For these next couple scenes, I'm switching camera angles to show you how to collect a sample in the field. To begin, take a photograph of the site to help track environmental conditions and weather patterns and make sure you're in the right spot. You can also pull this guy out to take air temperature. Once these numbers remain stable for about 10 seconds, you have yourself a final reading. To take water temperature and turbidity, simply turn this device on, place in the water, and read off the 2 measurements. The top one should give you the conductivity and the bottom should give you the water temperature in degrees Celsius. You can start with your turbidity bottle or your bacteria bottle as long as you remember which is which. To secure a bottle, press the tab located on this clip to open it wider. I'm starting with my turbidity sample, taken by using the larger reusable plastic container. Insert it into the clasp and cinch it down. Unscrew when you're ready to take your sample, and sample by dipping this several times into the river or water source from which you're collecting. Because the turbidity bottle is reused, it's important to make sure that there's no lingering residue inside the bottle.
To account for this, we'll take an extra precaution and use what's called the rule of 3, pouring out the first 3 samples before keeping the 4th.
Cap, remove, and place inside your dark, cool lunchbox. The turbidity sample is helpful, but the bacteria sample is arguably even more important. To reduce the risk of contamination, make sure that when you unscrew the cap, you're immediately ready to take your sample so that you don't risk airborne contamination by leaving the bottle uncapped for an extended period of time. When you're ready, unscrew, dip, cap, remove, and toss it in the lunchbox.
So that's it for the field part of the procedure, at least. So let's take a half a step back and review where we are. We need to record 6 things while we're in the field. And again, those 6 things that we need to do are to take a photo and write down any site observations that we see. We need to record air temperature, water temperature, conductivity, and we need to take 2 samples, a turbidity sample and a bacteria sample. My personal preference is to get those easy ones out of the way first, but you all can do it however you want. Split up the tasks between you and your partner however you like, as long as these 6 things are done before you all hop in your cars and turn around to either your next site or to come back to the lab. When you get back to the lab, you'll find that it's a little bit faster to start by processing your bacteria sample rather than your turbidity sample, because there's a step in the process that requires about 20 to 30 minutes of wait time. To begin the bacteria procedure, you need 5 things: a Sharpie, your bacteria sample that you obtained in the field, the small bottle full of ColiScan Easy Gel. This is that amber liquid that you took out of the freezer to thaw before you left the lab. You need a Petri dish, and you need a sterile pipette. Start by labeling your plate with the date and your site location, which is particularly critical if you test at more than one site. Once your plate is labeled, you can open your pipette from the bulb end. Carefully unscrew the top of the bacteria sample that you collected from the field, being careful not to touch the tip of your pipette to any unsterilized surfaces. Fill your pipette to the 3 milliliter mark by fully suppressing the bulb and getting that water drawn as close as you can to that horizontal line just below the bulb of the pipette. Once you have 3 milliliters inside your pipette, carefully unscrew the top of the brown ColiScan solution. Slowly squeeze your 3 milliliter sample of fluid inside the small plastic vial of ColiScan solution. If you squeeze it too hard, you'll run the risk of making your solution too frothy. We want a nice mixed sample, but we don't want to shake this and introduce too many bubbles to the mix. So to stir, simply swirl your wrist for about 30 seconds. The last thing we have to do for now is to take our mixed solution, to pull the top off of our Petri dish, and to slowly and carefully pour our solution onto the bottom part of the plate. Now the top fits over top, and the bottom, it's a taller piece that's been treated with a chemical that will allow this solution to solidify over the next 20 or 30 minutes. Set your sample aside somewhere safe where it can solidify undisturbed. While our bacteria sample is setting, we can move on to turbidity. Each kit has a black box, our turbidity meter, used to measure water turbidity, how cloudy it is. And inside, we have vials that are labeled blank. This is theoretically a perfectly clear liquid. We have a vial labeled 10 NTU, that is 10 turbidity units of water murkiness, as well as several unlabeled and empty vials that we'll use to take the turbidity of our actual water sample. And we can start by doing exactly that when we transfer our turbidity sample into these unlabeled glass vials. Because the glass vials are reused, we need to once again remember the rule of 3. And so it's easy to do this step by a sink so that you can pour out the 3 samples that come first before keeping the 4th. The other piece of equipment is, of course, the turbidity meter itself, this console right here. At the top, under the label that you can see right here is labeled WK4. That's our sampling well where we'll put our vials, but we can start by turning the machine on by pressing the button in the lower right corner. Now, our turbidimeters are extremely sensitive machines, and so we have to remember before we get a reading each week that these machines have to be calibrated in order to ensure that our reading is accurate. Calibrating the machine is something that is initially hard to remember until you get these 4 simple steps down on lock, and those steps are blank, 10, blank, sample. Once again, that's blank, 10, blank, sample. So let's start by measuring our blank. Now, the instrument is extremely sensitive to changes in light, and so any kind of smudges on these vials could cause problems. It's important to remember to give them a quick wipe with a Kim wipe or a Kleenex, some sort of soft tissue, before placing it with the vertical bar right here, facing forward toward the front of the machine, into your sampling well and back. And remember to close the top of the sampling well. Select Measure, and then Turbidity with Blank, and then scan your blank. Next, we want to do the 10 NTU sample, making sure again that that vertical bar is in front, making sure again that we give it a quick wipe with a Kim wipe or another soft tissue before placing it in the same orientation in your sampling well, facing forward and closing the top. Select Scan Sample and give the machine a quick second or two to spit out a reading. Once you have a reading, pan down to Calibrate and select it, and then toggle up or down as needed on the left keys until you're exactly at 10. Select Calibrate and set calibration. Now we need to run through this one more time with the blank to reset the machine with the new calibration, orient it in the same way. It's already been polished. Close the sampling well and select Scan Blank. Very nicely done. We've calibrated our machine. We're ready for our last step, which is actually obtaining a reading from our water sample itself. We haven't used this vial yet, so be sure to give it a quick wipe with a Kim wipe. And again, orient it in the same fashion, close the well and select Scan Sample. You got yourself a turbidity reading. You can take this number and write it down in your manual and upload it to our website for the world to see. I want to point out that in this test, today I'm using tap water, and so 0.37 is a pretty low number. Typically in the field, you're going to be seeing numbers that range between 5 to 15 and 20, and sometimes on particularly high water days, maybe as high as 40 or 50 units of turbidity. But once you have your reading, go ahead and turn your machine off, latch it shut, and toss it back in your bin for next week. At this point, you can revisit the bacteria plate that you've made. When you're very sure that the agar has solidified, you can go ahead and tape it shut, making sure that the label still shows, or label the tape when you're all done with the taping part of the procedure. When you're sure that the tape has secured the plate, you can go ahead and flip the plate over to prevent any extra condensation from forming and getting in the way of your field of view before slipping it inside of your incubator to incubate for 24 hours until you come back to check on Friday. So at this point, you're all done for Thursday. Just make sure that incubator is on and make sure that lab space is clean, and we can revisit this on Friday. Compared to Thursday, Friday is a pretty easy day. There are only a couple things we have to do. We have to get a bacteria reading by removing the sample from the incubator and counting the number of purple and blue colonies that appear on the plate. Don't forget to take a picture of your plate and to turn your incubator off. And then we have to upload the data. More on that in a jiff. But the last thing that we'll have to do is just to clean up our lab space at the end of the day, Friday, as we get those results online. Basically, what we'll be looking at are indications of purple or blue coloring, sometimes even a little bit of teal, indicates that you're looking at an E. coli rather than a regular bacterium. So here we can see anything that starts with a 1 is E. coli. We have another row of E. coli right here, so those correspond to these 3 blue colonies, as well as this array of purple ones at the top. If you see anything that's pink, or anything with these weird diffuse boundaries, if you see colonies with halos, those are signs that you're looking at something besides E. coli. And do keep in mind as you're looking at these colonies that we also have a bacteria key that's provided in the manual. And a little bit of practice here. This is a pretty ordinary looking plate, and a lot of people are tempted to look at a plate like this and to want to count E. coli where there are none. In this particular example, we're looking only at pink colonies. There's no visible E. coli on this plate. On this plate, we see 5 E. coli colonies. 3 have that pretty standard purple to bluish hue. A couple skew a little bit more teal, but they're all E. coli. We have 5 colonies on this plate. And a couple of tips of the trade here. Do be sure you're checking the sides of your plate. Sometimes if there's any motion as that agar is solidifying, some of these colonies can be sloshed up into the sides as well. So a couple more plates for you to take a look at. We can start with the one on the lower left. It looks to me as though there are 3 colonies here. 2 are very apparent, and the third is there, not quite as easy to see. There's 1 at the top right here. A second one right here, that's the harder to spot one, and then one more down here in the lower right. And in the upper right, count with me. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18 colonies in the upper right photo. But sometimes stuff goes wrong. These are both examples of plates for which we wouldn't be able to obtain a reading, and sometimes that happens. The upper right plate, both of these plates were probably mixed as they were solidifying, and so that has a tendency of stirring everything up and spreading those E. coli bacteria all across the surface of the plate. It's also possible that plate C may have been incubated at too high a temperature, so no E. coli bacteria were able to grow.
And while stuff like this is avoidable, it also happens from time to time. So do what you can to avoid it. But these plates are examples of plates that have gone wrong. The worst case scenario is a plate that looks like this. I believe this reading was collected from rockets landing directly across from Richmond's wastewater treatment plant after a stint of very, very wet weather. So if you see a plate like this, don't kill yourself trying to count each and every single one of these colonies. We cap our volunteers at 60 colonies per plate. And so if you can look and immediately tell that it's more than that, no worries. You can just write greater than 60 on your data sheet and you're good to go. I also want to briefly touch on the math. That's not too bad. That gets you from the number of colonies counted to a final bacteria concentration, a final answer that you can upload. And we'll actually ask for both of these data. But the state standard, keep in mind, is 235 colony forming units. So you can think of that as bacteria colonies per 100 milliliters of sample. That's a different volume than the 3 milliliter sample that we use when we collect this information from the field. So we have to convert. And we can do that by simply taking the number of colonies we have that we obtained from that 3 milliliter sample and by multiplying by 100 over 3 to convert to a concentration per 100 milliliters. And the last step is that we're going to round this up to the nearest whole number. And so as an example, if you have 4 colonies that you counted on your plate, when you multiply by 100 over 3, you get an answer of 133.3, which would round up to 134 colonies or colony forming units per 100 milliliters. Briefly, the online component here, if you haven't already, submit your schedules, but also go ahead and create a Water Reporter account. You can do that at waterreporter.org. There is an app that you can download for free. The account is free. And search for our group. What we'll be doing is allowing our volunteers who are in our group online to upload data. You can do that by going behind the scenes to readings. You can see this readings tab on the left side, and then there's a little plus sign where you can just add a reading, which will give you a sheet where you can upload all the information you need. And that's all I have today, folks. I just want to say once again that this program and our organization as a whole rely on the help of you all, our volunteers. And so I'm grateful for you all for listening to me for 35 minutes today, but also for all the help that you provide us year in, year out. We're very appreciative. And once again, I invite you to email me or call me with any questions. So thank you all again. Thank you so much for your help this summer, and keep in touch.